So in the lecture number 2 about modulation neuronal function by protein kinases and phosphatases, now we talked in the lecture 1 mostly about proteins, kinases that phosphorylate serine and threonine. Now we're going to talk a little bit about tyrosine kinases, and we're also going to introduce what MAP kinase pathway is because it plays an important role in receptor tyrosine kinases. We're going to introduce major phosphatases, which regulate, as we talked, phosphorylation, and then we are finally gonna wrap up and show how the signaling leads to gene expression changes. So protein tyrosine kinases phosphorylate tyrosine. Tyrosine phosphorylation, much less common, often serves as a recruitment of signaling molecules, adapter molecules, which have a domain, special domain called SH2 that recognizes phosphorylated tyrosine. There are two classes of protein tyrosine kinases. One very important are considered also metabotropic receptors, are receptor tyrosine kinases, which are transmembrane proteins and that have extracellular domain that will bind to the ligand, such as growth factor, neurotrophic factors, and then also they have intracellular catalytic domain, which can then phosphorylate tyrosines. And we also have non-receptor tyrosine kinases that are going to be cytoplasmic membrane associated kinases that are indirectly activated. So they can be activated both by receptor tyrosine kinases, they can be activated by G protein coupled receptor, they can be activated by any of the kinases that we have talked in the previous class. Now here's usually these receptor tyrosine kinases are close to each other, and then with ligand binding, or there is dimers already existing, so ligand binding will cause either dimerization or phosphorylation of each other, so autophosphorylation from another dimer in this another protein in this dimer. Now this phosphorylated tyrosine, as I said, will be recognized by SH2 domain in adapter proteins. Eventually these adapter proteins are going to bind to the downstream factors such as RAS proteins. Now RAS proteins are also GTP binding proteins. They're very similar to G proteins that we talked about in G protein coupled signaling. And then these RAS proteins are going to trigger the protein kinase cascade that will lead to many different downstream signaling. In this case we have MAP kinase pathway, which we're going to talk next. This SH2 domain can also be recognized as adapter proteins that can activate PI3 kinase, phosphatidylinositol kinase. This kinase is going to phosphorylate phosphatidylinositol 2-phosphate, that we thought is the substrate for phospholipases C-beta. PI3 kinase lead to additional phosphorylate, so instead of being phosphatidylinositol diphosphate, now it's phosphatidylinositol 3-phosphate, which then can activate directly the kinase called AKT. It's also known as PK-beta, and it's one of the kinases that's really crucial for the survival of the cells. It's very important for the neuronal survival. In addition, this phosphorylated tyrosine and the receptor tyrosine kinases can lead to the activation of PLC gamma. In this case, so PLC beta was activated for the PCRQ signaling or GQ signaling. Now PCL gamma is going to be activated through receptor tyrosine kinases, and it can also produce IP3 and deacylglycerol, and then do all the downstream activation from these two second messengers. Now, what are mitogen activated protein kinases or MAP kinase cascade? You must have heard about it sooner or later. You will if you haven't so far. It's a really kinase cascade in which one protein kinase phosphorylates and activates the next protein kinase in the cascade, so it goes downstream. It has this funny name, so you have MAP protein kinase or mitogen activated protein kinase, which will phosphorylate MAP protein kinase, which will go to MAP protein kinase. And then they will eventually, downstream, as shown here, will lead to the activation of transcriptional factors. And here you have all different stimuli that can lead to the activation of MAP kinase signaling pathways. So we can either have the receptor protein tyrosine kinase that we talked about, that can, through the RAS, lead to activation of ORAC pathway, but also stress response is going to activate JNK pathway and P38, which are two different MAP kinase pathways, which can result in different or altered response of the cells and altered gene expression changes in these cells. And this is again another illustration to show that combination of receptor tyrosine kinases and MAP kinase pathway will eventually lead to the alteration of gene expression. In addition to receptor tyrosine kinase, we have non-receptor tyrosine kinases. They are expressed throughout the CNS. There are many actually classes of these. So here are examples from two classes from SERC family, which are SERC-FIN, YAS, and alcycanolene. There's also YAC family, in which we have several YACs and TIC proteins. You don't need to remember these names, but they're actually really important. They're activated by any of these other kinases that we have talked. So they can be activated MAP kinase cascade, any of the G protein activated kinases, or they can be also activated through the cytokines such as leukemia inhibitory factor, ciliary neurotrophic factor, and interleukin 6. They are again very similar like the other kinases. They have this linker domain and they are activated by phosphorylation in the catalytic domain that activates them, because that's where they have also regulatory subunit is below, and phosphorylation of regulatory subunit is inhibitory. So they need two events to occur. One is to have phosphorylation of the catalytic domain to be activated, and also they need regulatory domain to be dephosphorylated for their full activities. These kinases in adults can play a role during development. They are really important from neurite outgrowth and differentiation. In adults, they play a role in ion channel activity, which we have shown illustrated in this panel here. For example, we have SERC activation. When SERC phosphorylates MDR, as you can see on the panel on the right, it's going to lead to the opening and more glutamate going, more effective glutamate through these, and more current going through these NMDR single channels. You can see that it's more often open, and therefore you have a larger current amplitude. Now, all of the things that we have talked so far, we talk about different kinases that can phosphorylate either serine, threonine, or tyrosine to change the structure of these proteins and then affect either their enzymatic activity, their degradation, or their interaction with the other proteins. Protein phosphatases are the molecular switches that undo what kinases create, pretty much. So they will hydrolyze this phosphate bond, phosphester bond, to give inorganic phosphate and then dephosphorylated protein. But it's really interesting, there are much less number of protein kinases, phosphatases than kinases. It's at least half of the number. And so there is a limited number, so they have a little bit broader specificity than the protein kinases have. They're classified based on their structure and the way that they remove the phosphate groups and the way they're regulated. They can be classified in two big groups, such as phosphotyrosine phosphatases. So these are phosphatases that remove phosphorylated tyrosine, and they're also phosphatases that remove phosphoserine or threonine. And they're also dual specificity that can remove from both tyrosine and serine or threonine. As far for serine or threonine phosphatases, we have they're called PP1, 2, and 2A and 2B. Some of them are regulated, and we'll talk more about it. As you can see, they have big catalytic domain, and for some of them, that's pretty much majority of the protein. They have a small inhibitory domain, and they can be regulated either through the binding of inhibitors to the inhibitory domains. They can be regulated by metal because they need some of the metals for their full activity, or they can be regulated by calcium calmodulin binding, and we are going to mostly spend time talking about these. Just two of these as an example of how they're regulated. Receptor tyrosine phosphatases come again in three different flavors. Ones that have dual specificities, and some of them, P10 as we have shown in the panel before, can actually regulate the phosphorylation of the NMDR to regulate the currents. They also have these very interesting transmembrane receptor phosphotyrosine phosphatases. They're really interesting because their activity is then regulated by binding of different ligands in the extracellular medium. And then, of course, they have some soluble that are going to dephosphorylate phosphotyrosine in the cytosol. On the other hand, we have pKa phosphorylating inhibitor 1 in this case, which again is the analog of hormone DARP32 in the other tissue. So DARP32 is going to inhibit phosphoprotein phosphatase to maintain this phosphorylation of potassium channels. What's really interesting, the other phosphatase that we're going to be talking more of how it's regulated is calcineurin, or PP2B. Calcineurin is regulated through increase in calcium level, which leads to its activation. So activation of calcineurin through the calcium, through, for example, an NMDA receptor, is going to then dephosphorylate this inhibitor and then activate phosphoprotein phosphatase 1. So we have the opposite effect from dopamine signaling and NMDA receptor signaling. So what is PP2B and calcineurin? We just talk a little bit about it. It's actually one of the most abundant protein in our brains. It has two subunits again. It has regulatory subunits and catalytic subunits. Regulatory subunits has two binding sites, from calcium commodulin binding site, which binds the calcium commodulin, and also it has a binding for its other subunit, which is calcineurin B. Calcineurin B is very similar to calmodulin, and just binding of it to calcineurin can lead to certain activation. And we have here again different signaling, intracellular signaling that is regulated by PP2B. We have again on the far left, we have this change in inhibitor protein 1, which, as we have talked, calcineurin PP2B is going to dephosphorylate to activate PP1. We also can see that PP2B or calcineurin will directly dephosphorylate either NMDA receptor or GABA receptors to change their internalization. We also can see that at the endoplasmic reticulum, a receptor for inositol 3-phosphate, we have phosphatase 2B, which can then act on the activity of these receptors. And also we have that in the nucleus, actually, a calcineurin can go and then it will change the phosphorylation of CRAB, changing the amount of gene expression changes caused by signaling that leads to the signaling from the membrane surface that leads to changes in gene expression. So this again leads us to the next step. So we have talked again, we have neurotransmitter binding to the receptors, and we have activation of GPCRs, G proteins, which then leads to production of secondary messengers. Second messengers will activate protein kinases, and again phosphatases to regulate the amount of phosphorylation. And this signal can go all the way to the nucleus, as we have just mentioned, that you can regulate the gene expression. So again, we start from the G protein, we have change in the second messenger, calcium deacylglycerol CMP, which will lead to protein kinases, which will then change phosphorylation. Now, there are some pre-existing transcription factors, such as CRAB and JUN, that we have briefly mentioned. Then they can then lead to the transcription of immediate early genes. So whenever you have signaling, we have these immediate early genes, and CFOS is one of the most commonly used in our system to look at immediate early genes expression. But these immediate early genes are also transcription factors that can bind to DNA and induce the transcription of RNA. So how does this work at the lab? It's represented here. And this tata binding box is usually 20-30 base pairs from the start site. And we will have accumulation of tata binding protein, which will recruit polymerase 2, and all of different factors that are required for its activities. This is a complex of RNA polymerase 2 complex. It has to come to that spot. But apart from that, you also need to achieve efficient initiation to produce significant transcription rate for transcription really to occur. And that is usually done by these other regulatory elements, here presents activator 1 and activator 2, which are further upstream from the start site or from tata binding box as well. Now, these regulatory elements, these are all cis regulatory elements because they're present on the same DNA sequence from which gene is transcribed. Activator 1 and activator 2 sites are usually palindromic sequences to which different transcriptional factors are going to bind. And as presented in the top panel, these transcriptional factors, either their binding or their activity once they're bound, it can be regulated by kinases. Kinases can also regulate the translocalization of these transcriptional factors from the cytosol to the nucleus, so they can regulate this activity in 3 ways. They can limit their localization to nucleus, or they can increase their localization to nucleus. They can change their binding to the DNA, either increase or decrease it, and then can either decrease or increase their activity. So usually at these activator sites, these response on these regulatory elements or proteins bind at in the complex, either they're dimers of their own homodimers or they interact with the other. We have here shown in the bottom panel, we have Cre response elements, which is CMP response elements, which respond to CMP signaling from GPCRs. They bind the protein called CRAB. Now, CRAB is always bound there, so in this case, phosphorylation is not going to affect the binding. What phosphorylation by pKa of CRAB is going to do, it's allow it to bind adaptive proteins. Now, what this does, it allows the looping of the DNA. So we'll have CRAB binding CBP that will then bind to the RNA polymerase II complex that's sitting at the Tata box. Most of these adaptive proteins, what they actually do, they're going to be in enzymes. And for example, here shown CBP, which is CRAB binding protein. CRAB binding protein is histone acetylase. Once bound here, it's going to lead to acylation histones and opening of DNA to allow the efficient induction of transcriptional to occur, to have a significant transcription rate, so the polymerase will be able to transcribe proteins, the RNA. So what is present on this slide is to show you that this transcriptional regulation by multiple signaling pathways is going to converge on the CRAB protein. So we have here what we have talked so far, CMP, and we have also in the slide before, is pKa activation is going to lead to phosphorylation pK of CRAB, but also activation of ACT, which we talk is going to be through the growth factors, through activation of PA phosphatidylcytose kinase. Then we also have kinase 2 that we talk with some of the isoforms of kinase 2 activated by calcium chromatin can go into the nucleus and lead to the activation of CRAB proteins. And also the MAP kinase signaling pathway can lead to the CRAB activation. Now what is illustrated here is that phosphatases as well can come into the nucleus and dephosphorylate CRAB to make it inactive. And this experiment is really interesting because it shows you something that you shouldn't be doing. It shows the difference between mass learning versus distributed learning, or mass training here because this was done in flies, or distributed training. So pretty much what this says, when you do mass training, when you try to cram as much knowledge as you can one night before the exam, you will CRAB phosphorylation through different kinases that phosphorylate to be active, but you will also have a lot of PP1 in the nucleus that's active, and therefore you're going to limit the amount of transcription induced by CRAB that again downstream leads to learning. On the other hand, when you distribute the training, you will have increased activation of PP1. You will have increased phosphorylation of CRAB because PP1 will be more inactive in this case. So it will not lead to significant activation of PP1. Therefore, you will have a better learning and a better memory of the things. This can be reproduced in the system. They were actually overexpressing inhibitor 1, which we talked about, can lead to the inactivation of PP1. And in that case, even though they had mass training, once they add inhibitor 1, they were still able to achieve the same level of gene expression, the same level of learning as when they had distributed training, but this time with the mass training. So here's a problem for you to think at home. This is introducing 3 different inhibitors you can use that inhibit different phosphatases. So cadaic acid inhibits predominantly PP2A. It is also tumorigenic a lot, but instead of the previous one that we have talked that it leads to PKC activation for ablator, this cadaic acid is tumorigenic because it inhibits PP2A, so it doesn't allow this signaling to be turned off. On the other hand, we have PP2 inhibitor cantharidin that is kind of inhibiting much more PP1 than PP2A. And then finally we have cyclosporin, which is very specific inhibitor of PP2B or calcineurin. Now you're looking at the protein X, and you know that activity of that protein X, which is expressed in the hippocampus and the cry for learning, is regulated by both phosphorylation and dephosphorylation. And what you have acquired, you have made actually antibodies that recognize total X proteins or protein, and also X protein is phosphorylate, so you have 2 different antibodies. Now I want you to think about the experiment that you would do, or series of experiments. You will determine which of these phosphatases, whether it's PP1, PP2A, or PP2B, that are dephosphorylating X. And that's it for today.